All right. Hi, everybody. This is the transcription and translation lecture video that goes along with the transcription and translation note sheet. So I'm going to just start the slideshow and we will get rolling. All right. So up until this point, we've talked about how DNA needs to be replicated in a cell for a couple of reasons. One of the reasons is that it needs to make a generational copy of itself. And whether that's like in your sex cells, like the gonads, or whether that's in like just the somatic cells that are that pretty much populate the rest of your body. So those are the two reasons is like DNA needs to replicate itself in order for that new replication to get passed on to future generations, whether it's going to be a sperm and egg or whether it's just going to be like another skin cell. But we're going to focus now on the other aspect of DNA. DNA, let's move on to the next slide. DNA's other function is that it is a repository of information on how to make proteins. So DNA basically is the cookbook for how to make a bunch of different kinds of food products. These food products can do all kinds of different stuff. And so maybe a good way to think about it is like DNA is basically like robotic blueprints on how to build a bunch of different robots that can do all kinds of different stuff around the cell. And so some of these robots, these proteins that can get made in the body, let me just make sure I'm not missing anything on this slide. Oh, yeah. Okay. So proteins are going to have many, many different functions in the body. And so let's talk about some of those functions. One of those functions that you're already familiar with from our module 2 is that proteins can be like enzymes, or not be like, but proteins are enzymes, or enzymes are catalytic proteins. And these are proteins that can break down or build up other molecules while they themselves don't get changed by the reactions. And that's a lot from unit 2 or module 2. The other type is that the proteins themselves, which are a sequence of amino acids, as we know from unit 1, the proteins, these sequences of amino acids can be folded and turned into a structure that aids like a skeletal element in the cell. So it could be a structural element like the cytoskeleton, or it could be something outside the cell, like in your fibroblasts, which are a type of cell that grow soft tissue. The extracellular matrix, or the scaffolding outside the cell, is what is your soft tissue. That's your connective tissue. And so that's a type of protein that they're making, a structural protein. Another type of protein that we talked about briefly in module 1 was transmembrane proteins. And so these are proteins or complexes or enzyme complexes that are actually embedded into the membrane of a cell, sort of like you can jam a toilet paper roll through the wall of a car or something, or the door of a car, and you have a connection from the outside of the car to the inside of the car. So these transmembrane complexes are proteins that are embedded in the membrane of the cell that help to transport things back and forth. They can help to take a signal from outside the cell and transduce that signal to inside the cell. Transduction of a signal without actual having the messenger enter the cell. So think about that. So it's sort of like if somebody rode up to the gates of a kingdom and said, "Hey, the British are coming," or something, and then the guy at the gates like, "Oh, the British are coming," and he goes and tells the king. The messenger doesn't actually enter the cell, and so that would be the same as these transducing of signals across the membrane. There's enzymatic reactions, so there's enzymes embedded into membranes, which are made up of protein. Not the membranes, but the enzymes are made up of proteins. They can help glue cells together, kind of like like your skin cells are super tight, watertight. So that would be the proteins that are joining adjacent skin cells to each other. And then also they can serve as ion pumps and active transports and things like that. So those are transmembrane complexes. Another type or function of a protein, and this is like literally, guys, this is why I'm spending like 20 minutes talking about this stuff, is that these do everything in your cell. The reason that you can function at all, period, have any level of self-conscious awareness at all, has to do with all of these proteins. And so these are basically like the scripts of life. So anyways, these hormone signals and receptors are types of proteins that can act like a signal molecule. So it can go through the bloodstream and interact with a receptor, which is another type of protein. And the receptor receives a signal, just like your antenna in your car receives the radio waves. Except in this case, the radio waves are proteins, and the antenna is a protein. So it's a protein interacting with a protein to transmit a signal. The last type of protein is one that we're familiar with from Unit 2, and that would be pigments. So things like chlorophyll, things like what else? Chlorophyll, beta carotenes, the xanthophylls, melanin, like in your skin, that makes your skin darker if you have extra melanin. So those are all pigments. Those are all proteins that capture certain wavelengths of light. All right. So why are we talking about this? Well, one of the reasons is that DNA has this, it has a transcendent property, meaning that you can take DNA out of one animal or one organism, and you can take that sequence of nucleotides, which is what DNA is. You can take that and you can supplant it in another organism, and that other organism may or may not, but it may express that gene. It might make that gene like we see here. So this is why genetics is so cool, is like we can snip things out of other organisms and put them in other organisms and make cool things like tobacco plants that glow, or pigs with glow in the dark nose and hooves. All right, so what is a gene? We know that a gene is a specific sequence of DNA nucleotides that codes for a functional product. And so that functional product could be a polypeptide, or it could just be the RNA itself. And we're going to talk about that a little bit later in this lecture, about the different functions of RNA. But for the most part, we're talking about where was I? The sequence of nucleotides in the DNA that codes for a functional product is a gene. Okay. Now, some things to keep in mind here is that if you remember, the sequence of DNA nucleotides as they appear on a DNA polymer or DNA strand, the sequence that those nucleotides appear in is parallel to the sequence of amino acids, like those beads on a string of a protein. And that's important because the order and sequence of beads on a string is what dictates the overall shape, which is the overall structure, which dictates the overall function of the protein. Okay, hopefully that's clear. All right, so we're going to talk first about the process of transcription. So this would be the idea that DNA is sort of a repository of information. And a repository doesn't do much other than it holds on to the information. So the DNA is holding the information. In order to get the information out of that repository, you have to make a copy of the information and take it out of the repository. And so that's what RNA transcription is. It's taking a copy of the information on how to build a protein. So we need a copy first. Now let's talk about prokaryotes. Prokaryotes are in an interesting situation because they don't have a nucleus that surrounds their DNA. Their DNA is floating around in their cytoplasm. So transcription in prokaryotes takes place in the cytoplasm. There's no chance for that RNA strand that gets made to be chopped up and spliced and stuff that we'll talk about later. There's no chance for any modification of that RNA before a ribosome will translate that RNA right away. So ribosomes will translate the RNA right away. All right. Now in eukaryotes, it's a little bit different story. So notice that the purple blob is the nucleus of the cell. Nothing is to scale, mind you. So this purple blob is the nucleus of the cell. We have our double-stranded DNA in here, and we need to make a copy of that information in the repository in order to do something with it, to make a protein out of it. And so because it's taking place in this protected environment, the RNA can have some processing done to it. We can remove some of these lighter colored shaded things. Those are sequences of nucleotides. We can remove them from the recipe and then glue everything together. And then now we're at a functional product that we can either send out of the nucleus and get it translated, or it can actually become something else. And that's what we'll talk about later. Okay, so anyways, let's go back to the idea of the gene. And this is where we'll kind of jump in. So there's two strands to a DNA molecule. It's, you know, it's a double helix. It's two strands wrapped around each other, and those two strands are glued together by hydrogen bonds. Now the strands, because they run anti-parallel to one another, right? There's complementariness between the sequences of both strands, but the sequences they're not ordered in the same way. So for an example here, like you'll notice that on the bottom, the sequence is T, G, G, T, 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 G, G, C, T, C, A. Okay, that's one sequence of nucleotides. And the complementary sequence above it reads ACCAAACCGAGT. Now, the pattern, right, like the syncopated pattern of like single, double, double, single, 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 double, double, you know, single, 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 like that pattern of changing nucleotides, the pattern is the same, but what nucleotides are filling that pattern is different. So it does matter which side of the DNA we are focused on to get a copy of it. So consider this, right? There's two sides to a DNA strand. There's a template side, and then there's something that we call the sense side. And I'll get to that in just a second. Remember when I said that the order and sequence of nucleotides is parallel to the order and sequence of amino acid as it appears on a polypeptide? Notice the first three nucleotides on this RNA strip are represented by one amino acid. So it's every three nucleotides is paralleled to an amino acid. And then you go the next three, and that's parallel with an amino acid. So the order and sequence of nucleotides is dictating the order and sequence of amino acids as it appears on a polypeptide. And so that's how DNA is the repository of information on how to build the primary structure of a protein. Okay. All right, now here's this image is a little bit more clear. That's why I wanted to wait till we got here. So the antisense strand is one side of the DNA molecule. The sense strand is the other side of the DNA molecule. Now, here's the interesting thing. So DNA is written in deoxyribonucleic acid. It uses thymine instead of uracil. RNA uses uracil. But if you'll notice, it uses all the other same nucleotides that RNA uses. It uses adenine, it uses guanine, and it uses cytosine. It just uses thymine instead of uracil. But other than that, they're pretty much the same. Okay, so look at the two strands of DNA. Now look at the copy that is made from that strand of DNA. Look at the copy that's made in RNA. You'll notice that that copy in RNA looks a suspiciously like great deal like the bottom strand of the DNA right here. And so that is why we call it the sense strand, is that the strand of DNA that matches identically the strand of RNA produced, albeit thymine instead of uracil, but the sequence is the same. That's the sense strand, because this strand of RNA will then get read by a ribosome and turn each codon into an amino acid on a polypeptide. Okay. So what does that mean? That means that this top strand, if this is the sense strand, if this is the one that's virtually the same as the RNA strand, that means the top strand is the antisense, because DNA is anti-parallel. One strand is anti-parallel to the other. So this is the antisense strand. There is another name for this strand. It's the template strand. This is the strand that actually gets read by the RNA polymerase, by the enzyme that's making the RNA copy. It reads the template strand and it produces a complementary anti-parallel strand that is identical to the sense strand, albeit with uracils instead of thymine. All right, let's carry on just a little bit more. Bear with me here. All right, so the RNA polymerase is going to read the antisense strand. I've already said that. That's from that paragraph right there. It's also called the template strand. The RNA copy transcription, so the transcriptive copy, the copy that the RNA produces, looks identical to the sense strand, but with uracil. Oh, I said that almost verbatim. Okay, I'm glad I didn't miss any of that. Let's see. In order for RNA polymerase to find a gene, okay, we'll get to that in a second. This is just an image, a different picture, if you will, of RNA polymerase engaging with the process of transcribing a gene. So you have the DNA is in blue. The RNA polymerase is this big gray blob. It's reading the DNA strand in a 3' to 5' direction. And as it's reading it, it's creating in a 5' to 3' direction the anti-parallel complementary copy of that gene. So as RNA polymerase is traveling along this gene, it reads a thymine and places an adenine in RNA. Then it reads an adenine and it places a uracil next to the adenine. And it slowly works its way down the gene from a 3' to 5' end on the template strand, on the antisense strand, creating an RNA copy that looks identical to the anti-parallel copy of the DNA on the other strand, on the sense strand. Okay, moving on. So we're going to talk a little bit about this diagram. This diagram, you're going to see it on your test. And so I'm going to ask you some questions about it. We're going to talk about each thing and what it is on the diagram. I made this diagram clearly. Obviously, it wasn't done to any super, I don't know, set of specifications, but it serves a purpose. Once you understand what each element is, the diagram makes a lot of sense and it's pretty simple. It's not overly complicated. Okay, so in order for this RNA polymerase, which is shown by the letter E, now you can in your notes, if you'd like, kind of like write down E is equal to, and that will help you on the test. I don't know if I rearranged the letters or not, but the diagram is the same. Okay, so letter E in this diagram is the RNA polymerase. Now, in order for the RNA polymerase to actually find the start site of a gene to copy, to transcribe, it needs to be attracted to that region by the promoter region of the gene. So the promoter region, you can think to get people to go see a band play in concert, you get a promoter. The promoter's like, hey, come watch the band play. The same thing. The promoter region is like, hey, RNA polymerase, come transcribe this gene. Okay. The RNA, let's see, so the promoter region inside of it has transcription factors. Transcription factors are the part of the promoter region that do the attraction to the RNA polymerase. So the promoter region is like, hey, I'm here. Transcription factors are like, sweet, I'm going to do the actual recruiting. They come inside the promoter region. They call up, the fans, and they're like, hey, come on, we need RNA polymerase here to transcribe our gene. So you have to consider that there's a whole crap load of DNA in each one of your cells that has genes on it that you don't want to transcribe. Your heart does not need to make a liver enzyme. Even though it has all of the instructions in its nucleus to do that, it wouldn't benefit the heart, it wouldn't benefit the organism as a whole. In fact, it would be energetically wasteful, and that organism would likely perish from the gene pool as well. So that's why the heart does not produce liver enzymes. I digress. The idea here, though, is that any one of your cells can produce any of the enzymes of a totality that your body can produce. So the question is, well, then how does it know which ones to switch on for which cell? Transcription factors. So transcription factors come to the promoter region. They sit inside the promoter region, and they can either, this is the cool part about transcription factors, switch on the gene or silence the gene, keep it shut down. It's like a light switch, the transcription factor. Transcription factor lands on the gene or lands in the promoter region, flips on the lights. Fans are like, there's going to be a show. RNA polymerase comes in, transcribes the gene. Okay. There is an area, and so the promoter region is A in this diagram. Transcription factors are C in this diagram. And then there's this area B right here. And so there's an area in the promoter region before the transcription factor binding sites. So the transcription factors, just to be clear, are attracted to the DNA in a sequence-specific manner, meaning the active site on the transcription factors targets a specific sequence that is found in the promoter region for that specific gene. It's crazy. It's mind-boggling how complicated that is. But that's like part of the attraction of biology. It's like a never-ending riddle. Okay, so just before those transcription factor binding sites, there's an area of nucleotides that is very rich in thymine and adenine. You might say, okay, why is there a whole bunch of adenine and thymine repeats in this area marked by B? The answer is this is the Tata box. A's and T's, if you remember from our lab, A's and T's have only 2 hydrogen bonds holding them together. C's and G's, cytosine and guanine, they have 3 hydrogen bonds holding them together. Energetically, it's much more efficient to have a whole area that's just 2 hydrogen bonds, because you have to separate the 2 strands in order for RNA polymerase to bind to one of them, the template strand. So in order for RNA polymerase to bind to the template strand, the strands have to separate. To separate, you got to break hydrogen bonds. To break hydrogen bonds, you have to spend energy. So spend the least amount of energy to open the strands, to get the transcription factors on, right? To attract the RNA polymerase to start transcription, and there you have it. Okay, that's the Tata box. That's the area marked in B. We talked about why it's easier to separate. Once the Tata box is open, transcription factors allow transcription to begin. RNA polymerase binds to the DNA strand, as you see here. It's already bound. It's already copied TAC into AUG. That's our RNA transcript. So it's just going to copy. It's just going to go. RNA polymerase is very myopic. Its only job is if I'm bound to a DNA strand and I have access to nucleotides that can fuel my reaction to create this polymer, this RNA polymer from the copy of DNA, like make this copy of DNA. If I have the materials to do it and I'm bound to a DNA strand, I'm just going to go. Now there is a stop. There is a stop, and here's how it works. So the RNA polymerase is just going to copy down the strand, and it's going to get to a sequence, a specific sequence of nucleotides called the polyadenylation signal sequence. It's polyadenylation, read my own handwriting, signal sequence. So once it gets that polyadenylation signal sequence, which is a mouthful, that sequence tells the RNA polymerase to just stop doing what it's doing, stop translation. You might say, okay, what is this signal sequence all about? What's this polyadenylation signal sequence about? Well, polyadenylation signal sequence is a sequence of nucleotides that has 2 purposes. The first purpose is it tells RNA polymerase to stop transcribing. The second purpose is that sequence of nucleotides ends up inside of the RNA transcript that was being made, because the RNA polymerase reads the signal and then drops off. So the signal gets copied and the signal is embedded in the RNA, and that signal comes important. Now I'm skipping, we're going to get to that signal in a second, but I'm skipping forward. So these are all of the, what do you call it? These are all the definitions of the terms that we just went through. And so you can look those up if you want on your own. Okay, so now we're going to talk about what happens once the RNA polymerase has hit the polyadenylation signal sequence, and falls off. Okay, now you have an RNA transcript, and this is purely in eukaryotes. This is purely happening inside a nucleus, and we know prokaryotes, they can't control this. It's not compartmentalized, and so as soon as it's done, a ribosome translates theirs into a protein. But for eukaryotes, we got this beneficial reaction chamber called a nucleus, and we can modify the RNA transcript that's made. So in eukaryotes, once the RNA transcript is polymerized, it goes through post-transcriptional modification, a 3-part process. Okay. Remember, I'm going to get to that polyadenylation signal sequence. We're going to get there. Okay. It's in the tail of the RNA, right? Okay. First, a 5' GTP cap, that's guanine triphosphate. It's got a little bit of free energy there, right? A triphosphate is added to the 5' end of the transcript. That helps to attract ribosomes and initiate translation, to power the ribosomes to start translating it. Here's the polyadenylation signal sequence. So at the 3' end, that's the very end, the very last thing that the RNA polymerase has transcribed and the very last thing it reads before it falls off. At the 3' end, a whole bunch of adenines, adenylation adds adenines to the 3' end. creates this poly A tail. So there is a enzyme that reads the polyadenylation signal sequence and then just starts throwing on a whole bunch of A's onto the tail. And so it makes this poly, which means many, A tail that goes on the end of the RNA. Now, why is that important? We understand that you need to power the 5' GTP cap. Makes sense because you have to power translation. But why add all these like A's onto the end of your tail? Great question. So if the RNA is mature and ready to be translated into a protein, it's heading out into the cytoplasm. Viruses hijack our cellular equipment all the time. And viruses are really good at taking their RNA and making our ribosomes read it and then create their own robots that take over our cells and turn into little virus machines, replicate machines, I should say. So it's in our best interest that if there's anything floating around in our cytoplasm that's RNA, we should probably digest it as quick as possible. And so there's enzymes floating around in our cytoplasm that your body makes that eats RNA. And it's good. It's like in its own immune system. It's like RNA bad. And it'll eat the RNA, which is fine, which is why we put on all of these A's onto the tail end of it. And so they start eating it, it's no problem. But by the time they get to the important information, boom, we've already made our proteins and we're good to go. It's almost like we tricked our own cell into, it's like we're pulling a fast one on ourselves in the cell. Okay. And then finally, the third part of the post-transcriptional modification is that a spliceosome, which is made up of RNA, remember RNA has other functions other than just being information on how to build a protein. So it's made up of RNA and proteins, the spliceosome. So the RNA portions of the spliceosome recognize the RNA portions of a transcript, and they're able to bind to them. The spliceosome has enzyme parts to it that cuts the RNA, and it removes introns. That's what the parts that are getting excised or removed are called introns. And then that spliceosome glues the rest of that RNA transcript together, what's left of it, into one long signal. And that those are exons. And so you splice out introns, you glue exons together. That is really confusing. But I don't know, that's how I named it. So we deal with it. Okay. I'll show you what this looks like. You can see that we have an RNA transcript that's full labeled. We have an untranslated region. That's what UTR stands for. Don't worry about that. It's just not the start codon. It comes before the codon. Here's our 5' GTP cap. And then everything after the start codon is part of the protein. That's what we want to make. Everything after the stop codon is going to be the polyadenylation signal and then the poly A tail. So there's a whole bunch of A's tacked onto the end of this that holds the enzyme at bay that eats RNA long enough so that we can this information between the start codon and the stop codon and turn that information into a polypeptide. So here's an activity of a spliceosome. You can see that there's some RNA in the dark red here, and then the light red is what is getting cut out. And so the intron is getting snipped out by the spliceosome. The exon 1 and exon 2 are getting glued together. And then you've got this cut out or excised piece of RNA that is not going to be used in the final recipe. And that's what a spliceosome does. Okay, so if we're splicing out our introns are cut out, and then we cut out the introns, and then we splice the exons together. And so you get this coding region. Once that happens, that is what codes for the protein. Your poly A tail keeps the enzymes at bay that eat RNA, and the 5' cap eat ribosome to get started. Okay, so that gets us caught up to where there are many other types of functional RNAs. And so what we need to talk about, because part of translation deals a lot with these segments of RNA that are functional without getting turned into a protein. They are functional in and of themselves. They're innately functional. And so we have RNAs, certain types of RNAs that are useful to us. So we said once RNA has been transcribed, so once RNA polymerase, which is an enzyme, it's confusing. RNA polymerase is a protein, even though it's copying DNA into RNA, it's a protein. All right, we're going to continue. So Where was I? Blah, blah, blah. Okay, so once RNA polymerase transcribes the gene into RNA, that RNA can be modified and turned into a functional piece of RNA that never gets translated. It just stays RNA its whole life. And it can also get turned into another functional type of RNA that becomes ribosomal. I'm getting ahead of myself. Let's talk about these with the PowerPoints. Okay, first type of RNA, that's the one we just talked about, mRNA, messenger RNA. Okay, so this is RNA that's been spliced, exons have been glued together. It's got a 5' cap, a 3' poly A tail. That's messenger RNA. It's got a message, it's got information, how to build a polypeptide, gets sent to a ribosome, gets translated into a polypeptide. Okay, we know that one. Then there's tRNA, which is a type of transferring RNA. And the tRNA's only job, it's sort of, you can think about the tRNA being almost like a Rosetta Stone, if you will. The tRNA is what the ribosome uses to make sure that it has the correct amino acid for the codon that it's reading. The ribosome is reading the codons and it's translating the codons, the language of a nucleotides, into the language of amino acids. So the order and sequence of codons on your transcript dictates the order and sequence of amino acids, but there's a translatory process happening. The language of the codon needs to get translated into the language of the amino acid. And these tRNAs are funny little molecules that are able to read the codon and match the correct codon with the amino acid. So it's transferring, it's bringing the amino acid to the ribosome that matches a specific codon. Okay. It has and the part that's matching is called the anticodon, right? Because codon is 3 nucleotides going one direction. An anticodon would be the complementary 3 nucleotides going the other direction because they're anti-parallel. That's why they call them an anticodon. Well, I'll show you an image of this in a second. And then finally, the last type of RNA that we'll talk about, there's many other types of RNA, just FYI. This is ribosomal RNA, so rRNA. And so this is RNA that once it's created, folds up and becomes the large and small subunits of the ribosome. And that's what's so fascinating, is that if you think about the machinery at play here, you've got RNA. DNA is the repository. So DNA is sort of like the, once civilization has evolved enough to create libraries or repositories of information, they no longer have to orally transmit stories. They can store them in the repository. Okay, but anytime you want to go get a story out, you have to go make a copy of it, bring it out, and tell the story. Okay. The thing about RNA is that it's not just there as a copy of the story. The RNA is also part of the person reading the story. So it's crazy. So the ribosome is the one that's translating the story, right, for the audience. It's the one that's turning it into a protein. And so you have ribosomal RNA is the one that's doing the translating. It's reading the script that came out of the cave or the repository, the library. It's a piece of RNA reading another piece of RNA that is using RNA that's bringing the amino acids it needs to build the recipe of RNA. So it's reading RNA, it's made out of RNA. RNA brings it the amino acids that matches the RNA. So it's like judge, jury, and executioner. RNA is doing all of these things together. Now the ribosome has some protein elements to it, but what's crazy about ribosomes is that their structure is so conserved, meaning that it has zero flexibility for mutation. If you get a mutation in your ribosomal DNA that makes your ribosomes, if you get a mutation in your DNA for the ribosome, you're dead. It doesn't matter who you are. Failure to develop. Ribosomes are so conserved across nature that you can take ribosome from bacteria and it will work in your cells, even though they've been separated by 3.6 billion years. That's how conserved the ribosome is. Crazy. Okay, ribosome activity. So a ribosome is made up of 2 subunits. There's a large subunit, that's the top part, and it's made up of 3 compartments. There's a compartment that matches the tRNA to the codon. So the tRNA is these blobs with like the little circle at the end. So remember, it's a translating apparatus. It matches for that amino acid, it matches to the codon. So it finds the codon, it has an anticodon for that codon, and it matches that amino acid to that codon and brings it to this ribosome. The ribosome is the big green thing. So you got the large subunit up here. It's got the A, P, and E site. So the A site is where the tRNA enters and matches to the anticodon. The compartment that adds the amino acid that tRNA just brought to the ribosome, there's a compartment where that amino acid is then attached to the growing chain of amino acids, or the polypeptide that's being assembled by the RNA, or it's being assembled by the ribosome. And then finally, where the empty, notice there's no amino acid attached to this one, the empty tRNA leaves, that's called the E site, or the exit site. And we can see another diagram. This one is on your test. I'm just telling you that ahead of time. So make sure, or it's in a worksheet at the very least. I think it's on the test too. So what we have here is we have the tRNA, and it's the ribosome, the large subunits up here, the small subunits down here, and it's reading the mRNA. It's reading the mRNA, and it's bringing the anticodon for that codon to the ribosome, and then it's passing it to the P site. So it goes A, P, E. P site is creating a polymer. That's a good way to remember it. It's creating the amino acid polymer. And then you got the E site is the exit site. And so you've got these amino acids are stuck to the tRNA molecules. Each amino acid is specific for a codon. The ribosome is the room in which the translating the room in which the translation is happening. Primarily, the translating apparatus is these tRNAs because they have a codon and they have an amino acid. So this is your Rosetta Stone, if you will. Cool. All right, moving on. So let's talk about what codons are. So a codon is a sequence of 3 nucleotides, 3 nucleotides long. Codons are found only on mRNA that's going to be translated into a polypeptide or a protein. So codons are only important when we're talking in context of translation. Otherwise, who cares what the 3 nucleotides are? Okay. There are 3 positions in a codon, and so first, second, and third position, and it's really what you would imagine. So the first position can be G, C, U, or A. The second position can be G, C, U, or A. There's only 4 nucleotides to choose from, and the third position, same thing. Let me back up for a second. So let's take a codon that's like GAG. First position is G, second position is A, third position is G. Or another one, GAU. First position G, second position A, third position U. So notice here, what we do is we'll take a look at the first position. We'll say, okay, G, and then now I'm constrained to that quadrant of this chart. And then A, so I'm going to come down here. My second position is A. And then U is my third position, and I'm going to trace it out here. So GAU translates to aspartic acid, and that's how we translate codons into amino acids. All right, let's give you one with AUG. So when ribosomes start translation, they always start with the codon AUG. AUG, let's figure out what it codes for. So we'll come to the first position. What is the first position? A. Okay, now I'm limited to this quadrant. Second position, U. Okay, so now I'm limited to this quadrant. And then G right here. So A, U, G. And it's this M, and I trace it all the way out here. Methionine. So methionine is always the first amino acid in a polypeptide. Always. That's the start codon. That's what initiates translation. Although methionine can be found not just at the beginning, you can have methionine found throughout the polypeptide as well. So the ribosome is going to read one codon at a time until it gets to the stop codon. So for the codon UAG, let's see what it codes for. So the first position is U, second position is A, and then third position is G. Well, it says stop. So that's a stop codon. When you get to a stop codon, it doesn't code for anything. It basically tells the ribosome that it's done. Its job is done. It's finished the translative process. What ends up happening is a termination factor that is shaped just like a tRNA comes in, except it's not carrying an amino acid, comes in, it lands on the stop codon. So the termination factor has an anti-codon that matches the stop codon, but it acts more like a wedge, and it separates the top half from the bottom half, or the large subunit from the small subunit of the ribosome. Okay, so we can summarize this into steps. The tRNA, and this is translation, mind you. So the tRNA with the anti-codon for the start codon, AUG, is going to bind to the start codon of the mRNA in the large subunit of the ribosome. The small subunit of the ribosome comes underneath and kind of closes like the jaws on the mRNA strand. The large subunit binds around that tRNA and it starts translation. So it shifts a codon over. That moves the first tRNA into the middle column, which is the polyadenylation column, or the it's a polymer column. So it's making a polymer out of amino acids. The new tRNA molecules continue to bring amino acids to the ribosome as the ribosome moves down the mRNA and translates the sequence of codons into the sequence of amino acids on the polypeptide. And so we can ask the question, okay, well then where are these ribosomes found? Well, ribosomes are mostly found on the rough endoplasmic reticulum. That's why we call it the rough endoplasmic reticulum. But ribosomes can also be found in the cytoplasm, and that's both in eukaryotic cells and prokaryotic cells. So even if you have a piece of RNA floating around in the cytoplasm, if a ribosome finds it, likely it will translate it, if it has a start codon. Okay, so that's why the rough endoplasmic looks rough. And then this is really cool. This is the summary of transcription and translation. So starting up here, RNA is transcribed from the DNA into an RNA transcript. Then the RNA is modified in eukaryotes. So it gets exons, or introns are spliced out, exons are glued together. It gets a 5' GTP cap and a poly A tail. And then it leaves through a nuclear pore. The ribosome pops on and initiates translation at the 5' end of the mRNA. The ribosome reads the codons on the mRNA and assembles a polypeptide. The tRNAs are the ones that bring the correct corresponding amino acid to the ribosome. And then a termination factor is going to bind to the stop codon, separates the subunits, and then the process repeats endlessly until the RNA is degraded. And that, my friends, is a summary of transcription and translation. That's essentially what happens. And we're going to get some practice with that, with the worksheets that I've given to you guys. If you have any questions, feel free to reach out. All right, cool. Let me turn this off. And you guys have a great day. Bye.